share my screen. Okay. Can everybody see?
Yes.
Yes, sir.
Okay. Okay. And there we go. Okay. So if you recall, in the last lecture, after we looked at in general what DNA replication is and some features of DNA replication, namely that DNA replication is semi-conservative. DNA replication occurs in a bidirectional fashion from what we call as the origin of replication, uh, and also that DNA replication always proceeds in a way that DNA is made in the 5' to 3' direction, and that the process itself is semi-discontinuous. By looking at what these terms mean, etc., and after looking at that, we said the actual way the DNA replicates can occur in 4 kind of different ways, which we call as the bubble or I type replication, theta replication, uh, what we call as D loop replication, and rolling circle or lariat or sigma replication. Okay, and yeah.
Sir, are these the major types observed modes of DNA replication, or are these
Uh,
only the modes?
Actually, you are breaking up. I can't hear you. Can you say it again, please?
Sir, I said, are these the only mode of DNA replication, or there are new ones also, and these are the major ones?
There are other modes also, but these are the major ones. Okay. In nature, nature always throws you a surprise in terms of how DNA replication occurs. But these are the 4 main ones. But yeah, there are other ones which are many times variations of uh, these are slightly different than these, and there are other modes of replication. Okay, but by and large, the 2 main ones, okay, are actually bubble and theta. And then we talk about D loop and rolling circle because of the fact that we have mitochondria, and rolling circle is seen in many viruses, okay, which switch between linear and circular DNA and stuff like that. Okay, so these are the 4, but there are other variations of how DNA can get also replicated. Okay, so this is not the complete list. Okay, there are other modes. Okay, does that answer your question? Hello?
Yes, sir. Yes.
Yeah. Okay.
Yes,
So
sir.
okay. So looking at all of these modes of replication, now let us actually look at typically how prokaryotic replication occurs, and much of the information that we have as of today as to how DNA replication occurs is based on a lot of studies in bacterial systems. Okay, so what we are going to do because of in a sense the fact that the maximum information that we have as of today is by studying these systems, is we are going to look at prokaryotic DNA replication, and that too mainly what we call as a theta model. Okay, and after looking at the theta model, we will kind of look at eukaryotic replication or our own replication, and look at how it is slightly different from how it occurs in the bacteria. But irrespective of which mode of replication there is, typically a replication process will have 3 steps. Okay, what we refer classically as the initiation of replication, which is how replication actually begins. And the second is the elongation, that is a bulk process where the DNA actually gets replicated or duplicated, as you would say. And the last step, where the process of replication is stopped by the organism to move on to the next stage. Okay. So irrespective of whether we are looking at bacteria or higher order eukaryotes, or even if you look D loop and rolling circle, there are always these three steps: initiation, elongation, and termination. Okay. Now, with respect to theta model, or with respect to prokaryotes, or with respect to bacteria, where this occurs most of the time, uh, the process of DNA replication invariably begins at a specific site. Okay, and this specific site is referred to as the origin of replication. Okay, now if you remember, when we are doing recombinant DNA technology, and we were looking at plasmids, PBR 322 and PUC 18. There also in that plasmid, we mentioned this little spot, which we always said is the origin of replication. And that origin of replication is a place where the process of duplicating of the plasmid, in that case, or in our case, the duplicating of the circular genome of E. coli, in this case, begins. Okay. Now, typically, this origin of replication has got some unique properties. Okay, and in case of E. coli, for example, it is a stretch of around 245 base pairs. Okay, out of a total 4 million base pairs that E. coli has, this region of 245 is where always the replication will start. So in other words, the beginning of the replication of the circular genome of E. coli, or for that matter, many other bacteria, doesn't begin at random anywhere along the circular DNA, or in case of linear chromosomes, anywhere along the linear chromosome. There are always specific places where this replication will start, and those specific places are referred many times as the origin or origins of replication, in case there is more than one. Okay. Now, typically, what you will find, here is an example from E. coli, is that this sequence of 245 base pairs tends to contain some types of repeats. Okay, and in this case, there's a tandem array. Tandem means one next to the other of 313 mers. Okay, so there is a 13 base sequence. Okay, shown here in a sense like this: GA, T, then CT, then any nucleotide. N means any nucleotide, then TT, then any nucleotide, then TTTT. Okay, now remember, I've shown the sequence of only one strand. Okay, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Now you have to realize that if it's double-stranded DNA, the opposite sequence would have been C, T, A, G, A, any complementary nucleotide, then AA, complementary nucleotide, AAAA. Okay, and this sequence of 13 base pairs occurs 3 times right next to each other. Okay. Now, we'll look at how it looks in the next slide, but it is from this point that replication starts, and it goes in both directions because the replication is going to be bidirectional. Okay. So here is that slide. So here is that sequence that I just talked about. Okay. A tandem array of 3 13 base pair sequence. Okay. Now, what is very, very important to realize is that if you look at this sequence, see how many of this is AT, okay, relative to GC. Okay, now look at this. First one will be GC, second is AT, so that is 1, then it is TA 2, then TA 3, then TA 4, 5, and then TA. So at least 9 out of this 13 are ATs. Okay, and not only that, somewhere near that sequence is another sequence which is also rich in a sense in TA. Okay, so see 1, 2, 3, 4, 5, 6 out of 9. Okay, out of 80 sequences, and there are 4 of these. Okay, see that 4 9 base pair sequences, and this is supposed to be the binding site of this protein which is called as DNA. Okay, now, so at the origin of replication, you see this two features. One, repeat of 13 mer 3 times, of which 9 out of 13 at least is 80. And here 4 sequences which are not tandem but which are distributed, and also their orientation might be this way or that way. See this arrow, what they indicate is this sequence oriented in this way or exactly the opposite way, okay? But doesn't really matter. But there also, out of 9, 6 of them are 80, okay? As I said, this is the binding site of DNA A protein, which binds to here relative to this site here, okay? Is that clear? So once this DNA A protein sees this set of 13 base pair sequences, relative to that, it binds somewhere here where it also finds this very beautiful 9 base pair sequence, but which is separated, but which is not tandem. Okay. Now, what is DNA A protein? Okay, so look at it. DNA A protein is the one which recognizes the ORI sequence. ORI sequence means origin of replication sequence. So as I told you, this DNA protein is the one which recognizes, okay, this origin of replication sequences, and what it does is very interestingly opens the duplex at specific sites in this origin of replication. Okay. Now there are two ways to look at what we call as the origin of replication. What a lot of people say is this entire set of sequences is what we should be calling origin of replication. Okay. While some people say that kind of this is a recognition sequence, but this is where origin of replication actually is. Okay. Because this is the pair which is actually separated by DNA A protein. Okay. And the third group says, no, this is the origin of replication because that is what is recognized by DNA protein first. It really doesn't matter so long as you understand what is happening. What is happening is this protein recognizes these sequences, and then relative to that, it goes nearby and at very specific sequences opens up the DNA. Okay, and the reason it is able to open up the DNA very nicely and easily is because here the DNA is AT rich rather than being GC rich. Remember, AT rich regions have got only 2 hydrogen bonds between the two base pairs, while GC rich regions will have more base pairs. Okay, so it appears that this origin of replication is designed to be opened up a little easier than any other random sequence, because any other random sequence, if I take 13, half of it should be A, half of it to be GC, right? Probability. Similarly, here 9, half of it should have been 80, half of it should have been GC. But you notice one thing, there's a bias towards the sequences having more 80, which means this set of sequences are designed so that they can be opened up with a little less effort than what would have taken if they had been regular sequences or they had been rich in GC, which would have made opening them more difficult. So this DNA A protein, yeah. Hello. Yeah.
So the 13 base pair which you mentioned, so it is compulsory that sequence only, or it can be any sequence having more 80 linkages?
See, what you find is that this sequence which you see here, no, is almost bacteria species specific. Okay. So what you might find is different bacteria. Remember, see this, I put this as N and N here. What that means is this could be either GC or it could be CG, it could be AT, it could be TA, in the same way here. Okay. Now what is observed is that across many bacteria, when you look at all the promoters, it always tends to have a bias where there is more AT relative to GC. But what exact sequence it will be is many times dependent on which bacteria you are looking and many times which strain and which species you are looking. Okay. So once I say E. coli, then E. coli will invariably have this. Okay. In the same way, if I am talking Bacillus subtilis, it could be similar to this, slight variation, but it will still be biased towards having more AT. Okay. So as far as a given organism is concerned, this is always conserved. Okay, but it could be slightly different between different bacteria, but the bias of being AT rich still remains. Okay, does that answer your question?
Yes, sir.
So this is not the only
So,
sequence in nature. Yeah.
so
Hello.
is it to ease the sir? So is it to ease the interaction having more AT?
Yeah. The philosophy is that if it is richer in AT, there will be less hydrogen bonds holding that region. Okay. So that region is more easily separatable by an enzyme relative to a sequence which has got more GCs, which has got more hydrogen bonds. Okay. So maybe the thought is that the region, these are sites where replication can begin more easily, is because the enzymes are able to sit here and open up the sequence with a little less effort. Okay. Elsewhere, if you look at the DNA, you might find that the way ATGC base pairs are many times it's a 50-50 distribution, okay, on an average, okay, across a stretch. Okay? So,
Yes,
so
sir.
that is what DNA A does, okay. Now, in addition to this protein, there are a lot of other proteins which take part in the process of DNA replication. Okay, so I'm going to just list them here and tell you a little bit about them, and we'll see what they do in the pictures that come up a little later. Okay, then there's another protein called DNA B, which is nothing but a helicase. Okay, so what does a helicase do? It unwinds the DNA. Okay, and the reason you need to unwind DNA, remember that the way DNA replication occurs, it is semi-conservative, right? So the original strands open up, and then each is used as a template to make what is called as a complementary DNA. Then there is a DNA C protein which helps the binding of DNA B protein. Okay, so it says required for DNA B binding at the origin. Okay, then you have HU, what is called as a histone-like protein. Okay, and what does that do? It binds to DNA and it also helps with the initiation process. Then we have what is called as primase, which is also referred to as DNA G, which makes those little RNA primers that are required for DNA polymerase to add bases to as part of the DNA synthesis. Then we have single stranded DNA binding proteins, which are called as SSB or single stranded binding proteins. What do they do? When the DNA has been unwound and separated, there is always a tendency for the DNA to again base pair because the opposite strand has got all the requisite complementary bases, right? So even though I separated the two, if I let them go, AT will come together again form hydrogen bond, GC will come together again form hydrogen bond, and I do not want that to happen because I want to replicate that area. So these single cell binding proteins bind to this DNA when it is separated and do not allow it to again form a double helix, okay, so that replication can proceed. Okay. RNA polymerase, okay, that's also there, uh, but at this point, as part of DNA replication, it's a surrogate enzyme which just helps the DNA. Okay. Okay. Then you have DNA gyrase. Okay, what this does is it relieves the strain generated by DNA unwinding. Okay, now remember, what helicase does is basically just break the hydrogen bond and separate the DNA. Okay, now when the hydrogen bonds are broken and the DNA is separated, what that does is that it creates a strain both ahead of that region and behind that region. Okay, it's almost like if you had a uh, rubber band, no, and you started twisting it for a while, and you reach a point where it is highly strained. Then, if you ask somebody to put their hand in the middle of the rubber band and try to separate the two rubber bands, what you'll find is that region will separate, but because of that, the other regions will crunch up on itself because now they'll get overwound because of what is called as supercoiling. Okay, now I do not want that to happen because if that happens, my process of replication will be hampered. So DNA gyrase relieves that torsional strain of you opening up the DNA so that later on DNA replication get stopped because of jumbled up DNA in the front. Okay. And what does DNA methylase do? This DAM, okay, DNA methylase. What it does is it methylates the sequences at the origin of replication of the parent strand. Okay, it is not written here, but it does it of the parent strand. Okay, so what it does is see the sequences GATC sequences here. The DNA methylase will methylate the sequence prior to the process of replication actually beginning. Okay, and it does so for a reason. Okay? So very quickly, if I were to go one by one, I have a protein which binds to the origin of replication and opens it up. Then I have a protein with the help of another protein which unwinds the DNA. And then we have a histone-like protein which also helps in that process. We have an enzyme which makes a primers. We have some proteins which keep the separated strand stabilized. We also have gyrase which relieves the strain of unwinding, and the parent's GATC sequence at the origin of replication are methylated by this enzyme called DAM methylase. Okay. So just to give you an idea of how exactly this happens, here is the picture now. Okay. So if you look at this region here, this is double-stranded DNA, and if you look closely, you can see the helical structure of the double-stranded DNA. So this is the region where there are that 13 base pair repeats, which are 3 of them, and then there is this 4 9 base pair repeats. Okay. Now see what happens is DNA A comes in along with ATP, and remember this region could also be supercoiled, because remember normally DNA is nicely packaged around the histones so that it can be stored in a small place. Okay, so that background supercoiling is always there. Now what does DNA protein do? Along with ATP, based on recognizing this region, it then takes this region here and wraps it around itself. And as it is doing that, it slowly unwinds the region, which is this 9 base pair region. Okay, you can see that. And as it does that, other proteins B, C, and your helicase will start coming in. Now that essentially is that point which is called as the origin of replication, from where you'll find you'll get replication in either direction, one in this direction and one in this direction. Okay. Now as this is happening, remember DNA B, C, and DNA. Okay, B, C are the ones which unwind the DNA. But as this is happening, other proteins also come in and you start forming those RNA primers. Okay, that's why they say that in this process, opening of the strand occurs and also priming occurs. Okay, and once the primer has been added, then the actual process of replication begins. And as the process of replication begins, then these things simply fall off. Okay, this DNA A, B, C are no longer needed now. They just fall off. The only thing that remains, in a sense, is helicase and your topoisomerase and your SSBs, which keep the strands separated. Okay, so DNA A kind of goes away after initiating this process, and then only helicase and topoisomerase and other primases and all, they stay. Okay, but what exactly happens at this point to the DNA primer, or a little later to DNA, which is, let us say, composed of N nucleotides, deoxy nucleotide triphosphates are added systematically one at a time in the 5' to 3' direction. Okay, and this is done by DNA polymerase. Okay, so if you look at this slide here, all the proteins are mentioned, but there is no mention of DNA polymerase, because we are only talking about the process of initiation and not the process of elongation. So after initiation occurs and the primer is added, that is when the polymerase starts to come in. It will start adding deoxynucleotide triphosphates, forming a phosphodiester link between the 3' OH and the 5' phosphate here, and then the DNA will increase by 1. What will be added will be dependent on what is the sequence in the template, and will be exactly complementary to that. Okay, and then if you remember, we already talked about this in the rDNA class. The polymerase itself has several requirements. It needs a template, it needs a primer, it requires all your deoxynucleotide triphosphates, it requires magnesium, and it also requires other accessory enzymes, which we'll look at a little later. Okay. Again, I'm shown this just like before. If there are n bases, a deoxynucleotide comes in, lengthens the DNA by 1, pyrophosphate is released, and this breaks down to 2 inorganic phosphates, and that is what drives the polymerization reaction by making this reaction highly delta G negative, okay, or thermodynamically favorable. Okay. More specifically, if you remember this picture also, this is the DNA strand which is already sitting there. This is the 3' end. This is the new deoxynucleotide triphosphate which has come in. This OH attacks what is called as the alpha phosphate. Okay, this is called as the alpha phosphate, this is the beta phosphate, this is the gamma phosphate. It attacks the alpha phosphate, releases the beta and gamma and pyrophosphate, and this base will then join to this. And growth will occur in the 5' to 3' direction. Once this is added, this OH will attack the next nucleotide again at the 5' position on the alpha phosphate, releasing pyrophosphate. Okay, and that is how the DNA strand grows. Again, another cartoon here. Okay, here showing that role of magnesium. Okay, see that magnesium, how it helps by binding to the phosphate. So here we have this DNA strand. Here is a template. Based on the template, the complementary base has come here. The complementary base is a deoxynucleotide triphosphate. See that? OP, OP, OP. It binds somewhat like this. Then this OH attacks this alpha phosphate. This pyrophosphate is released. And I get the joining of the strand. Okay? Shown in a slightly different way. Again, all trying to tell you the same thing. See, this is a template. I've shown that in the 5' to 3' direction. This is the growing DNA strand where relative to T, A has been added, relative to C, G has been added. Now there is a free OH here. Now the template has A, so deoxynucleotide triphosphate of thymidine comes in, and then this OH attacks this alpha phosphate, releases a pyrophosphate, and now T is added to G, and I get the extended DNA where correctly T is bound to A. Okay, again shown here.
So,
Here is a base. Yeah, yeah.
So then what happens in the case of lagging strand when there is a breakage and Okazaki fragments come into picture?
Okay, yeah. We'll come to that. But so what I'm trying to tell you now is look at the different ways I'm showing the same reaction, which is how DNA polymerase extends the DNA in the direction of 5' to 3 by 3' OH attacking the alpha phosphate and releasing pyrophosphate. What base comes in is dictated by the template. Okay. Now, very interestingly, when people were looking and trying to find out and isolate and study this polymerase which is doing this job, okay, this enzyme which does this is called as DNA polymerase. In E. coli, they actually found 3 polymerases, okay, and these 3 polymerases which are called as polymerase 1, 2 and 3 were discovered in this sequence. Okay, so the number 1, 2 and 3 are given because of the chronology of their discovery. Okay, so this fellow was discovered first, this fellow was discovered second, and this fellow was discovered third. And as soon as this fellow was discovered, people started getting very excited. Okay, yeah, we found that enzyme which is supposed to be the one which is doing DNA replication, and they started studying it, and they found that it has all the things that are needed for it to replicate DNA. Okay, it is a big protein. It has got a molecular mass of 103 daltons, or what we call as 103 kilodaltons. Okay, it has got the 5 prime to 3 prime, what is called as polymerization activity, but it also had what is called as a proofreading activity. So it could come back in the 3 prime to 5 prime direction and take out a piece of DNA. Okay, we'll see what that proofreading means. It also had a nice polymerization rate, about 60 to 20 nucleotides per second it could add. And it could add up to 200 at a time, before which it would simply fall off from the template and stop, and then maybe another polymerase would come and start from that point. Okay, but then at that point, the excitement of discovering the polymerase more than compensated for the fact that some of these properties are not properties which are actually suitable for replication of E. coli. Okay, now think of this. E. coli ka doubling time is how much? Approximately 20 minutes, right? On an average, 15 to 20 minutes. So if I want to replicate my DNA and there is 4 million base pairs, okay, and I have to make basically a strand that big, and if I'm adding 20 nucleotides per second, that will be 200 in 10 seconds, okay, 200 into 6, which is 1200 in 1 minute, and then that would be 12,000 in 10 minutes and 24,000 in 20 minutes. How the hell am I going to replicate 4 million if I'm going to replicate at this speed? Okay, that 20-minute doubling time is impossible to achieve if I'm going to add nucleotides at this very small rate. Okay, but at that time, a lot of people, realized, maybe there might be some other mechanism and stuff like that. And it went on for a while till they discovered this polymerase. Now look at this polymerase 3 relative to polymerase 1. Okay, this has got one subunit, molecular mass of 103 kilodalton. Look at this one. They've got more than 10 proteins which come together to do this polymerization, and its size is about 9 times this, 791 kilodalton. Okay, or 791, 7,091,500 is the molecular mass. Okay, it also has proofreading capacity. What it doesn't have is this very unusual activity, 5 prime to 3 prime activity. Okay, and in a sense, therefore, it might appear that it is got less properties than this. But actually, for DNA polymerization, this activity is not required. And I'll tell you why this is required a little later. But see this part. This is very important. Its rate at which it adds nucleotides can be up to 1,000 per second. Okay. Now we are talking, an enzyme which can potentially do replication fast enough so that you can actually replicate the entire genome of E. coli in a time that fits with its own doubling time. And not only that, look at this other activity, processivity. How many nucleotides it adds before it leaves or it dissociates? That is more than 500,000, which means what? This is a polymerase which once it begins the process doesn't leave. Okay, so in Hindi I call it this lage property. Okay, एक बार चालू किया तो वो लगे रहता है. I'm going to add a polymerase which needs to replicate the entire genome. You don't want it adding a few and leaving, because the minute that happens, replication will stall, and then you have to wait for another polymerase to come. Okay, so as of today, it is now known that the main polymerase which actually does DNA replication in E. coli is not the one which was discovered first, which is DNA polymerase 1, but it is actually DNA polymerase 3. Okay, so what is the role of DNA polymerase 1? Okay, DNA polymerase 1 plays a role in DNA replication, but it plays a role in mainly removal of that RNA primer and replacing it with DNA. Okay, remember when we talked about how DNA synthesis occurs, we said every time you need a small piece of RNA to start things. But at the end of the day, I don't want that RNA to be part of my DNA, and that removal is actually done by a DNA polymerase 1. So what does 2 do? It takes part in repairing any damage to DNA. So we'll study about that when you look at DNA repair. Okay. So as far as replication is concerned, the bulk of the work is done by 3, and this one basically does work at the level of primer removal. Okay. So see this. So I'm now talking about 3 now. The bulk of DNA synthesis is carried out by this polymerase. It has got extremely high processivity, which means that once it attaches to the template DNA and starts adding nucleotides to form polymer, it will not come off till it has done most of the job. The important part is it has got a very high rate of nucleotide addition. Okay, high enough to then realize that I could have a E. coli replicating in 20 minutes flat. Okay, it also has a very complex composition, more than 10 proteins which come together to do the job. Okay, but this is very important. It's got a very low error rate. It has got high fidelity. Okay, now when in this COVID scenario, all of you are online, and we are mostly using Wi-Fi. Okay, Wi-Fi means what we call as wireless fidelity, which means whatever information I am telling you, as it is being passed from my computer to the tower, through my router, to your tower, to your router, much of this information is traveling without wires. Okay, and in spite of it doing so, you are listening to exactly what I am saying. Okay, and that type of fidelity, or what is called as correct reproduction, is called as wireless fidelity. Okay, now I am using the term here high fidelity. That means simply I am saying that its rate at which it looks at the template, adds the correct complementary base, it does it so accurately that it makes only one error per, see this, 10 million nucleotides. Okay, which is 10 raised to 7. Okay. In fact, in reality, if you look at the fact that there is also DNA repair systems which work together at the time of DNA replication, when E. coli DNA is replicated, the error rate is even lower than this. It is 1 in 10 raised to 9 rather than 10 raised to 7. Okay. Now, the reason for this low error rate is this 3' to 5' activity. So, as this DNA polymerase 3 looks at the template and correctly adds the complementary base and does the polymerization by 3' OH attacking the 5' phosphate and so on and so forth, it also always makes sure when it is ready to add the base to a template, what is the scenario of the previous one. Okay, so for example, if it sees a AC by mistake, when it is ready to add the next base pair, which is T, and it says, "Arey, this is not right," then it simply stops this, goes back, removes the C, then adds the correct T, and then it continues. Okay, so this going back, correcting, and then continuing is referred to as the proofreading capacity, and that is also called as 3' to 4'. So it's very interesting, right? So this enzyme normally adds very fast in this direction, but whenever it makes a mistake, it just does a backspace, puts the correct alphabet and continues. Just like when you are texting, you are doing something and you realize you have made a spelling mistake, you very simply go backspace, change it and continue forward. So this does something very similarly, and because of that, it has got a very high fidelity rate, or what do you call it, very high accuracy rate, as it is adding these bases at this high speed. Okay, and the speed is how much? Close to 1000 nucleotides per second. Okay, and how does this polymerase look? Okay, see this. A lot of people, if I talk about it in a lightly philosophical fashion, or maybe a spiritual fashion, DNA polymerase almost looks like a hand. Okay, is shown here. See this, the palm of your hand. And a lot of people, even scientists of repute, have sometimes used the word that the polymerase appears to be like the hand of God. Okay, where it is perfectly designed as if somebody is taking your DNA very nicely and replicating it. Okay, and here is all the activities. This is where pollination occurs. This is where the proofreading activity is. And this is not one protein. Remember, there are close to 10 proteins which come together to form this structure. Okay. And as the DNA is being replicated, the fingers and the thumb come together to push the DNA along the protein as it is doing the replication. Okay. There is another picture where DNA is bound to the polymerase. So what you see here is DNA. This is empty polymerase, and this is a space where your double-stranded DNA comes in after it has been separated to a single strand. So the template comes here, and then that DNA is looked at, it is read, and you add the complementary base pair. Okay. So now coming to the question that was raised earlier. Okay, when synthesis is occurring in the 5' to 3' direction, if I were to look at any replication fork and I put in this condition that I can add bases only in the 5' to 3' direction, I realize that this creates a very tricky situation for me. Okay, for example, these two strands are separated here. I add a little primer, which is shown here, a little dark blue here, and then based on that primer, I'm able to continuously add this. And the reason I'm able to continuously add this is because I'm moving in the 5' to 3' direction. And not only that, this DNA is being unwound and replicate and made free in this direction, okay, by helicase and your topoisomerase. So as my DNA is freed and the orientation of this strand is actually 5' to 3', I'm able to make my complementary in the correct 5' to 3' without stopping at all, because in front of me the DNA is constantly being separated. But as I'm doing this, what about this other strand? On that strand, I cannot work in this direction, because if I do, that will be against the way the polymerase works. Direction would be 3. So I'm in a sense forced to do the synthesis on this strand in bits and pieces, where as soon as here I add a primer and start synthesizing this in the, then as I move forward here, again I add another primer and continue in this direction. I go again forward, I add a third primer and continue this. In other words, this is occurring in a continuous fashion, this is occurring in a discontinuous fashion, and that's why collectively this process is called as semi-discontinuous. Okay. Now remember, last time I told you that this terminology semi-discontinuous sometimes appears to be a little weird or a little odd. Okay. So to clarify that, this is what we would mean if it were continuous. Let us say the fork is going like this, and I'm able to synthesize one strand and the other strand at the same time without stopping at all. Okay. So in both, as the fork is moving, one is made in the 5' to 3', other is made in 3' to 5'. That I would call technically a continuous replication of DNA. Okay. What would be semi-discontinuous? Okay, semi-discontinuous is what we just saw, where one is done continuously and other is done in the stop and start. And what would be discontinuous? Where both strands are made in a stop and start fashion. Okay, if that were to occur, I would call it as a discontinuous process. Now, because here one is done continuously and the other is done discontinuously, I use a terminology as semi-discontinuous to indicate that there is a portion of DNA replication which actually occurs without stoppage, and there is a portion of DNA replication which occurs in a stop-to-start fashion. As opposed to these two, where here it occurs without stoppage in either direction, here it, in both strands it is stop and start. Okay. So is everyone comfortable with this terminology? Why we say
Yes, sir.
semi-discontinuous? Okay. So that now you
Sir,
realize, yeah. Hello.
sir, then yes, sir.
Yeah, yeah.
Hello.
Hello, hello.
So
I can hear you.
doesn't it create a time disparity
Okay.
because of the
Uh-huh. Okay.
yes sir that
Yeah, I know what you're saying. Okay. You're saying that here I'm making things continuously here in one direction, and then suddenly I have to actually go back and go in that direction. And because of that, there might be what we call as a time differential. Okay. Not only a time differential, it appears at the moment very troublesome, no, for this to occur. Okay. मैं ये डायरेक्शन में मस्त जा रहा हूँ, बीच में घूम के ऐसे जाना पड़ रहा है. Okay. Now this polymerase is very elegant in the way it does it. Okay, I'll show you how it does it, so that in a true sense, there's almost no time lag. Okay, and see what it does, and it appears almost like a genius way of evolution, and therefore maybe the terminology hand of God. Okay, we'll come to that in a second. Okay, see the proteins which are present at the replication fork. Okay, we saw the proteins which are present at the initiation. Okay, now see the proteins which are present during the process of elongation. Okay, you have single cell binding proteins. Why do we need that? We need that because the strands are separated. I don't want them to rejoin. I still have helicase for DNA winding. I still need a primase. Why do I need primase? To keep on making the primer on that Okazaki fragment. I need polymerase 3, which is the main enzyme which does strand elongation. I need polymerase 1. Why I need? To remove the primers and fill the gap with DNA. Then I need ligase to do the ligation of the Okazaki fragments. And I need gyrase to prevent a tangling. Okay, I will show all this in the next cartoon. So if you look at the replication fork, which previously I shown you in a very simple fashion. This is continuous, this is Okazaki and stuff. Actually, it is very crowded. Okay, so here is a leading strand, and look at this polymerase here. Okay, this is a polymerase here. What is it doing? It is adding more strands. Okay, and here is a fork. What are these? These are single cell, single strand binding protein. Also primase, which is adding the primer, so that I can do this. This is a work. After that, polymerase 3 continues to add polymer, and after it reaches this point, it goes away, and polymerase 1 comes. What does polymerase 1 do? It removes this primer, okay, this RNA primer, and fills it with DNA. Okay, that is what it has done. It is going to fill this with DNA, and after it is done, there will be a little gap, and that gap will be sealed by ligase. So now that is complete. So means that this Okazaki fragment, which we talk about, as short pieces which are made in a discontinuous fashion, after a while will all be joined together and all will be full DNA. And why will they be full DNA? Because periodically polymerase A will come, remove all the RNAs, fill it with DNA, and ligase will come and seal it. So after a while, you'll find that this will also be completely DNA, although it has been made in a discontinuous fashion. Okay. Now again, that issue that this is going to go continuously, while this is going to be stop, start, stop, start, stop, start. Okay, and and that would create maybe a time lag. Okay. Now before we do that, no, remember that if this was the origin of and the two forks are going in either direction, and what happens the hand side fork, right? With the left hand side fork, see the leading strand is coming like this and the lagging strand is going like that. But in the other fork, it will be exactly opposite. This will be the leading strand and this will be the lagging strand, which is in the 5' to 3' direction, opposite to the direction of separation. Okay. So in the two replication forks, the leading strands will be diagonally opposite and the lagging strands will also be diagonally opposite because of the different orientation and the directionality of unwinding. Okay. Now, as I said, this is what is done by polymerase 1. This is the RNA primer. As DNA polymerase comes synthesizing the previous Okazaki fragment, it reaches this point, it goes away. Then this polymerase comes and fills it up with DNA, removing the RNA. And then there's a little gap there, and that gap is filled by DNA ligase, and you'll get now a full strand. Okay. This is again shown here. Okay. So I have this strand which is going in this direction. I have this other strand. Primer is made. I got helicase B, single cell binding protein. Gyrase is here. The strand is kept separated. Then I make this Okazaki fragment in the other direction. Later on, it will look somewhat like this, and it then be sealed. Okay. Now, this is where, actually speaking, the beauty of replication is seen. Look at this next slide. Somebody asked a question that I'm making this in this direction while I'm making the lagging strand in the other direction. That creates both a time issue and in terms of processing issue. Okay? Now see what actually happens in real life. This is one of the strands in which the orientation is 5' to 3'. Because of that, I can make a primer here and I can start my synthesis of leading strand, and that will go continuously in this direction because my replication fork is moving in this direction. Now see what the polymerase actually does with respect to the other strand. It actually doesn't do synthesis in this direction. Okay. What it does is it takes this lagging strand, okay, a template, and twists it. Actually does a little turn. When it does that turn, see what happens? The orientation of 5' to 3' ends up like this, rather than appearing to be like that. So what happens now is after I make the primer, I actually start adding to the template in, relatively speaking, the same direction as I'm doing this. Okay, and see how I manage this. There is this little what you call as link protein to which two molecules of polymerase are added. Okay, are attached, and both actually are working in the same direction. The only difference is one is adding in a continuous fashion, one is adding a discontinuous fashion, but the directionality is same, and the reason it is possible is because I have twisted the strand in such a fashion that 5' to 3' appears in this direction. Correct? Now, sorry, think of this. If I left the strand like this and I was adding a primer here, then my 5' to 3' would be in this direction. Okay. But if I turn this strand around like this, then my 5' to 3' is actually in the same direction as it appears to be happening here. Now you see what has happened because of that? That issue of time, the issue of having to turn back and do things is taken care of, because what I do is I don't turn back, I turn the template so that it is twisted in the correct orientation so that I can move in the same direction as this is moving, although I do it in a discontinuous fashion. Okay, is that clear? Somebody was asking me how we are able to
Yes,
manage that lagging strand. So So the beauty is this. So the other strand is turned around by this enzyme, because remember there are 10 of them. So they work together to turn this around so that I do not have an issue of turning back and doing it in the other direction. Then what I do is simply I add this, then I let go of this strand, take more strands and I turn it around, and so on and so forth. So there's a leave and catch, leave and catch, so that I'm always doing the lagging strand in the correct orientation relative to this. Okay, and that's how my replication will continue till I reach the point where I'm kind of diagonally opposite to where I started. Remember, if I have circular DNA and I start at the origin of replication and I move in either direction, initially the shape I will get will be theta, correct? But as I go along that theta, I will reach a point where both the replication forks are kind of 180 degrees apart, or at 180 degrees to where I started. And that point will be where I have to actually stop the replication. So if I go back very quickly to the theta model which was here. So if I start my replication, let us say at this point, and I start going, my two forks are moving in this direction. So as I go along, my theta will appear. At one point, the two replication forks will be exactly opposite, which will be at point C relative to point B, which is the origin of replication. And at that point, I have to somehow stop the replication, because I would have replicated my entire double-stranded circular DNA. Okay. Now, how I do that will be the topic of the next lecture. Okay. So we are at this point now. We have added the leading and lagging, and we have gone all the way and probably reached the diagonally opposite point from where I started. And I'm ready to now stop the replication because I have actually replicated my entire DNA. Okay. Now think of the rate. Now this can add at 1000 per second. This can also add at 1000 per second. So in the replication fork in this direction, I'm adding 2000 per second. Similarly, if I'm going in the other direction, I'm also adding another 2000 per second. So totally the rate of addition is 4000 nucleotides per second, which is 40,000 per 10 seconds, which is how many per minute? .4. Okay. So now I'm talking speeds which are enough so that I can have a DNA E. coli replicating in close to what is called as 20 minutes. Okay. So any questions before I stop sharing my screen?
Yes, sir.
Yeah.
So can I speak?
Yeah.
Sir, I had a doubt with the lagging strand orientation. If we are changing the orientation the way the leading strand is, and the polymerase does the activity in 5' to 3' end, then why does the strand is broken into fragments? Why doesn't it go continuously?
See, you have to remember that when this strand is made in 5' to 3, right? This I technically can never make continuously in 5' to 3, because if to make it continuous in 5' to 3, I have to start somewhere here. My primer should be here. But the problem is this DNA is still double stranded, no? So I can never make this strand in the 5' to 3' direction continuously. But what I have done is to help me not turn and do this. Okay, see, the problem here is if I'm moving in this direction and I have to make this strand in this direction, essentially my polymerase has to turn around and move like this. Okay, so which means my polymerase has to be very oddly placed, one going here, one going here. And since I'm making this in bits and pieces, how many polymerases I will need? That is always a question. So what I do is I use only two polymerases. One which is associated with this strand making in this direction, and one which is associated with this strand. But what I do is, rather than making this in bits and pieces, by going back and turn this around. So when I turn this around, what happens is, essentially, this polymerase is also appearing to move in this direction, but it is actually making in the 5' to 3. So what it does is, it does up to this point, okay, and till it reaches this. Then what it does is, it lets go of this strand and then pulls more which have been separated now, and again turns it around and then adds in this direction. So the direction of addition is still 5' to 3. The only difference is, I am not turning back and adding. I am adding in the same direction, but I am still adding in a stop to start fashion. And the way I do so is by pulling this, adding this, letting go. Again, pulling fresh strand, adding in this direction and letting go. So the advantage is, as these fellows are moving in this direction, there is no need for this fellow to turn back and go in that direction, and that would create a technical problem as far as the complex is concerned. So are you getting the point? I can never make this strand in 5' to 3 continuously, simply because of the way the two strands are oriented of DNA, because remember, they are anti-parallel. But what I can do is help myself and not having to turn around and do the other strand. So the lagging strand is actually also physically made as if everything is moving in the same direction. Okay, that is the only difference. Okay, does that make
Okay, sir.
sense to
Thank you.
you?
Yes, sir. Yes, sir.
Yeah.
Sure. Thank you, sir.
So you have to think through what exactly is happening here. It is not very easy to visualize. Okay, and that's why you feel, okay, why can't I make this also continuous? Because I'm moving in the same direction. See, I'm moving in the same direction, but look at the orientation. See this strand. This is 3' going like this, going like this, going like this, going like this to being 5'. Correct? Now look at this. Yeah, this is 5' and I'm going like this, 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 and now I reach this is 3'. Correct? So now
Yes.
look at this section here. This is 3' to 5'. This is actually 5' to 3', but this part is 3' to 5', like this. Are you seeing? Are you seeing what I'm saying?
Yes, sir.
So
Yes, sir.
this is actually having the same orientation as this. That is why, because I just turned the strand around, and therefore I am able to move in the same direction. Okay. So this is 3' to 5'. This is 5' to 3', but at this portion, the orientation is exactly the same as this time. Okay. And that is the trick that this polymerase does. Okay. Now, if you go to YouTube, there are videos which are animation of how this polymerase works. Okay. And that nicely shows you how it lets go, catches again, lets go, catches again, so that this lagging strand can also be made in the same direction. Okay. Does that clarify your doubt?
Yes, yes, sir. Thank you so much.
Okay.
Sir,
So,
there are some doubts on in the chat box.
okay. So let me stop my sharing here so that I can get to the chat box. So here is the chat box. So let me see. So the role of magnesium is just to stabilize the complex while addition of deoxy ATP. Yeah, magnesium plays the role of stabilizing that ATP because of the negative charge that those phosphates have. Okay, so that's the main role. Okay, then let us see. How does DNA enzyme identify the origin while the DNA is still supercoiled? Okay, if the DNA is coiled, there are chances that origin of gene is hidden by the coils around it. Okay, yeah, you are right. But what happens is, when packaging of DNA occurs, okay, something very interesting is done. The packaging doesn't occur at random. What you find is that places where DNA needs to be active, especially what we call as origins of replication, or what we call as start sites for transcription, they are very systematically packaged so that they are not buried inside the supercoiled DNA. Okay, so as part of this process of supercoiling and part of packaging of DNA, care is taken by the cell so that it doesn't bury sequences which need to be accessed as part of either replication or part of transcription. Okay, so and therefore those sections can be searched by the DNA when replication signals are given, and it will find it. Okay, and that is what actually happens. Okay, a very good question though. Okay, Aditi, very good question. Okay, so did I answer
Thank you,
that
sir.
question, Aditi? Yeah.
Yes, sir.
Okay.
Sir,
Yeah.
and I also had a doubt in DNA gyrase. While uncoiling, you said that
Yeah.
it relieves the strain that is created by unwinding of DNA. So I
Correct.
just
Correct.
can't imagine kind of how it happens. The DNA is still circular at the beginning, and if
Uh,
we are and if we can imagine the DNA is opening, so
mhm.
the strain is kind of what to say travels to the more coiled region. So how is it relieved?
Ah, see what topoisomerase does, okay, or what we call as DNA gyrase, no? Another name for it is topoisomerase. What it actually does is, when the strain is seen, it actually makes a cut in the DNA, okay, and it cuts the DNA and allows one strand to be passed around the other strand, okay, and after it does that, it reseals it. Okay, so you can imagine now that if I have double-stranded DLA, which is helical, and I pull it apart, and because I pull it apart, at points both ahead of it and behind of it, the DNA will start getting crunched up. And the way I relieve the strain there is by cutting one strand, allowing it to go along around the other strand and putting it back. Okay, and that is how this topoisomerase 1 works. Okay, so it cuts one strand, turns it around the other strand and reseals it. That's why it relieves the strain. And there's another enzyme which is called as topoisomerase 2, which is even more interesting. What it actually does is very systematically cuts both the strands of DNA, allow one strand to turn for a little while, and after turning, rejoins it. Thereby, it relieves the strain. Okay. So you see these two enzymes, they relieve the strain by actually cutting and rejoining the DNA, and that's the only way you can relieve the strain. Because remember, the DNA is circular, right? So either side se strain aane laga na, so there is no place for it to relieve the strain. But if you had linear DNA, no, life is a little easy. When you have linear DNA, what happens? The end simply starts to turn around, and when the ends turn around, that strain is relieved. Okay. Uh, so topoisomerase are actually very interesting enzymes. So relieve the strain by actually cutting and rejoining the DNA. Does that make sense now? How the strain is
Yes, sir.
relieved?
Thank you.
Uh,
Yes.
Actually, I have an enzyme which actually cuts the DNA, allows it to be passed around the other strand and rejoins it, and that's the way. Okay. And now the interesting part is this: when replication occurs, no, and I make double helical DNA, remember I have turned the DNA around and unwound it. So it is now in an unwound state. After replication, no, I need to do exactly opposite. I need to put that strain back. So many times I need the topoisomerase isomerase again to again get back my double-stranded DNA as I go further. Okay. So topoisomerase is needed for both relieving the strain and actually creating the strain when it is under strain, because stability is only achieved when the strain is perfectly balanced. Okay, is that
Sir,
okay?
so how it creates the strain?
So remember how it undoes the strain, it cuts and takes the DNA in one direction, no? Now if it cuts and takes it in the opposite direction, it will create a strain. समझा ना? तुम्हारे हाथ में
Yes.
समझो 2 रस्सी है, हाँ, और मैंने राइट हैंड साइड का रस्सी को लिया और उसको लेफ्ट हैंड साइड का रस्सी के ऊपर से डाल के नीचे से वापस पकड़ा, और ऐसे डाला और नीचे से वापस पकड़ा. Then I will create a strain of one type, right? Where one strand is going around the other strand in one direction. Now, if I want to unwind the strain, what do I simply do? I do the exact opposite. I take that strand and instead of moving in the original direction, I move it in the other direction and catch it again. If I do that, I will simply do the exact opposite of what I did before. So
So what we
copoisomer is
do?
this.
So we do a positive supercoiling type.
Exactly. So previously if I did negative supercoiling, now I'll do positive. Previously if I had to do positive, now I'll do negative. Okay. So all I need to do is cut and pass the DNA in either the clockwise fashion or the anti-clockwise fashion. In one direction it will release the strain, in the other direction it will generate the strain. Is that clear?
Yes, sir. Thank you, sir.
Okay. Okay, so shall I stop my recording and I will meet next time. But before I do that, no, let me go there and I have to download the attendance. So I have downloaded the attendance. Now I'm stopping the recording and